you guys were real quiet. I'm gonna start picking people or punching people for those you guys said good. But I'll start with what so Dr. McCary is interested in soil ecology. I too, since high school, known I wanted to study soil ecology, but I've actually often been on the periphery of it because I've found it somewhat unsatisfying. And the central problem for soil ecology is, how do you describe how 1,000 things are interacting? And so that's my question to you. How do you wrap your head around, it's one thing to have a spider and a grasshopper and a grass, and it's another thing to have 10,000 spider species, 5,000 grasshopper species, and 6,000 sources of food. How do you wrap your head around understanding that immense complexity, and do you have a vision for how we do that, how we move forward? What's a way we can make this tractable?
That seemed like a very unfair first question. It's supposed to be, like a softball. Okay. No, that is a phenomenal question, though, and that's something that, for me, I've been challenged with, and I've done it in several ways. I guess the easiest way I've done it is really just try to extract all the different invertebrates, arthropods. So there's techniques that we use. So it's we call it Berlese Tobin funnel. You put the soil on the funnel, it heats it up, and most arthropods are very sensitive to drying out, and so they kind of move across this heat gradient, and you have this extraction valve that allows for you to actually count what was in this sort of substrate of soil and litter in that case there. And you're right, you can't really sort of visualize their interactions, but what you can do is when you have a sample like that, you can see if there are correlations between abundances of certain taxonomic groups. And so if you have predators, you can kind of see how that sort of relates to the prey in that case. So that's a little bit more correlational in that sense there. And yes, it's not fully satisfying, but you can envision that you have many samples across many different sites, many different plots, and if you can see similar correlations across different trophic levels, one can envision that those are things that are real in reality. To get at the more mechanistic stuff, for sure, I have since moved into sort of microcosm experiments, and so we have these incubators in the lab that we have at Rice, and it's not really reflecting an entire community of predator and prey, but we've done some experiments looking at how the soil structure itself can really influence predator-prey dynamics. And so what we've done is kind of have different treatments of different soil types and kind of basically inoculated them with known numbers of prey, springtails namely, and also known densities of predators where we can actually count and see directly how they are sort of related to one another. And there's a paper that, a project that I'm currently hoping to get out sometime soon, but what's really fascinating is that the soil texture itself can really dictate how predator and prey interact with the soil. So usually predators are bigger organisms than they prey, and you can envision that the structure of the soil can dictate how they interact. And so soil that is have large aggregates, prey and predators can move more easily. And so you see a more interesting dynamic across predator and prey in that case there. But you have really compact clay-like soil where you have small pore sizes that only the prey can move through, but the predator can't. It basically decoupled their relationship in that case there. And so that's kind of one way these microcosm experiments that really tease apart those meaningful interactions in that case there. And then you have the whole issue of how you do that with microbes. And yeah, I just basically sequence stuff and then look for correlations at that point. So I actually haven't figured out how to do that more directly, but that is something that I hope to be able to resolve. And I have some ideas about that. I could spend hours talking about that, but that's kind of in a very, I guess at a very high level. Those are the approaches I've used to really understand those interactions.
Oh, people, you guys got to jump in, or else I'll keep asking questions non-stop.
Yeah. So is that something that you've used gradient forest for in the past, just something like that to kind of find the major environmental variables that are driving all these associations, or what do you typically look?
Yeah, no, that's exactly the case. So usually I'm looking for different either treatments or different levels of invasion of invasive plants and seeing how those, or urbanization, seeing how those various factors can change those correlations between predator and prey in that case.
Do you use genetics at all for the invertebrates as well? You were talking about doing that with like bacteria, but instead of like sitting and actually getting them to drop out of your sample.
Yeah, no, that's a great question. So actually, I have a project right now where we've actually done that, where we basically took whole samples of like plant tissue and soil samples as well, and you can kind of extract arthropod DNA, and then you can kind of get an understanding of like you similar to like the microbial stuff. You just get a database of like OTUs or operational taxonomic units that each one is supposed to indicate a separate species. The one thing about insects, especially soil arthropods, a lot of them are uncharacterized, and so you know that they're different, but you don't know what species it is, and so that's kind of the challenge in that case. It's something that I'm currently confronted with, but one of the things we've done, which I think is kind of cool but also challenging, is that we actually did the extractions on a number of soil samples, and we actually identified what they are. And then we took both samples, ground them up, extracted the DNA, and then we can actually match those OTUs with the actual samples that we identified in the lab. We haven't gotten to that point yet, but that's kind of the direction we're going in that case.
Can I follow up, I guess, with a question for both of you guys? So that same kind of question, how do you deal with complexity? I feel like you could ask that about any ecosystem, not just.
But it's still orders of magnitude beyond anything else that any ecologist does. You have,
Well,
not only do you have literally tens of thousands of species, but those species change within hours, like genetically and functionally.
so yeah, I mean, you're getting it. So there's techniques that we use to reduce complexity, that we start aggregating things. Well, these guys are similar. We'll deal with these at the genus level, we'll deal with them at the functional group level.
Yeah.
But that requires those feel good about that kind of aggregation or simplification. Maybe it requires a certain level of background natural history knowledge. Is that what's missing with soils? Does that make it harder to do that kind of lumping?
I definitely would say yes. Yes is the true answer. So I mean, when I kind of like yesterday alluding to all the different groups you have, I mean, like mites alone, the level of knowledge you need to be able to key those out to species, I mean, it's like a lifetime of work, to be honest. And you get like hundreds of thousands of different individuals, and it's like it becomes impossible to actually count them out and identify them. And it's actually very tedious because you actually have to mount them on a compound slide, look at the genitalia. And this is just like for one specimen. You may have like hundreds in one given sample. And so it becomes very intractable to kind of, to really get at it at that level. So I definitely take a more functional group approach where, you know, you can look at a higher classification and know that it's most likely a predator, most likely a fungivore. It's not perfect, but I think right now that's kind of where we are. But I definitely think that you do see some meaningful differences across these different taxonomic units, and that's something that I've been able to kind of push forward, and hopefully, yeah, maybe we'll have a more sophisticated approach where it can kind of get down to like the genus and species level. But I think right now I've been really thinking about like a trait perspective, thinking about a higher classification to really get at.
I think the microcosm work is the way to go, because that gives you the overall answer. Like,
Yeah.
that tells you if something's important and if you can measure it at that ecosystem level. And in fact, there's a history of ecologists going to microbial and soil insect research, because you can have 50 reps on this table and you can do an experiment in a week, which we typically can't. So there's a terrific strength in working with soil ecosystems, but really diving down and understand. We don't even have a catalog of what is there. And the whole species concept doesn't even work. And so that makes it difficult. But yeah, like working with the microcosms, and I do plant cell feedback research, and there we use the plant as a bioassay of what the soils are doing. And that's satisfying, and it gives you an answer. But to try to figure out really what's going on and driving that, it's tough. So kind of following up on that, I'm curious about your impression. I tell people all the time, I think, 50 years, it's already started, but all farmers are going to be adding back to plant growth promoting bacteria, fungi, like we're going in that direction. We're going to get an understanding.
Yeah,
But kind of, do you see it being applied, and do you see there's also a big problem. There's lots of failed inoculation experiments.
100%.
It's hard to, like, you're at microcosms with changing the cell structure. That's a really cool approach, and it's nice that you saw that. But there's lots of examples where people have tried to inoculate something and then nothing happens.
Definitely.
So I don't know what you see for a future for like manipulating the microbial, either for experiments or for in practice.
No, it's a phenomenal question. It's one of those things that I'm often, I get this question a lot actually. And one of the things that the science is not there yet. I think we definitely, in an ideal world, it'd be nice to be able to find like the certain species or the context of where these species can be, can promote, crop yield or whatever you're interested in. But we have a long way to go. And I think part of it is because a lot of these species that, or if you want to think about microbial species in that way, a lot of them are almost impossible to grow in the lab. And so even if you take a soil where you try to isolate all the different bacteria and fungi, only a small proportion of that can actually be sort of grown in the lab. And so it becomes nearly impossible to really learn the function of some of these microbial species. And that's where you kind of really need to be able to get a more informed sort of evidence-based approach to promote growth. So if you know that this one species associated with this one plant is going to actually bolster growth, like it's really, really hard to get at that in some ways. And so we basically just doing darts in the dark right now on that. But I do think, coming up with some clever experiments that where maybe you can do some sequencing plus some lab work and just basically looking at different communities that microbial communities that seem to be, helping out in some ways. But I feel like it's really hard to do, and I haven't really figured out what's a good approach to get to that point. But the short answer is that I definitely think it'd be ideal to get to that point, but I definitely don't have the brain to be able to figure out how to do it the best way. One of the things some of my colleagues have tried is that if you have a community of soil microbes that seem to be healthy, you kind of take some water, saturate that soil, kind of make a sort of a soil slurry. You then syringe that community and place it somewhere else, and then you can kind of see if that's going to have the beneficial effect. But one can envision that's not going to be done at scale. And once again, you still don't know which one of those species are responsible for bolstering growth if you see that. So I think the short answer is like, yeah, it's nice to be able to do that, but I don't know if we anytime soon will be able to figure that out in a way that I think will lead to something that's really meaningful and actually gets closer to what you're trying to do. I don't know if you have those.
Yeah, so approaches I've taken working with the bioassay approach it's nice because when we sequence, we also have plant growth data. And so we can sequence and we can say, here's the microbial community of this plant, and here it is on this plant, and we know that plant grows well on this soil, poorly on this soil, and so we can make a correlation of, problem is you have a correlation with 5,000 OTUs, and you're like, which one of them is doing it?
Yeah.
And that's, but I think tying, like you said, having a plant growth response or an ecosystem, a CO2 response or a nitrate response, if you have some value that you're interested in manipulating and you can correlate that with the microbial community, I mean, that's the first step, just to try to prioritize who's doing what.
Yeah.
Something else along those lines I've been thinking about, and I'm curious to hear what you think is, we do these multivariate analyses on all the sequence data. And so you have this cloud of data, but I'm often looking at, normally we look at axis 1, axis 2, because that explains the most variation.
Yeah.
But then when I actually start to find correlations in axis 6, axis 7, axis 8, so it's these like rare things that aren't doing much that are actually correlated with plant growth. And it's got me thinking like, the structure of the soil microbial community, and like, I feel like there's just this massive organisms just cycling the carbon through, but they don't really have much to do with the plant growth or with nutrient cycling. There's a lot of functional redundancy, and I don't know if you have a sense of like what proportion of the community is kind of redundant and what's really driving down other parts of the community. That's a pretty vague question, or what really affects plant growth, you know?
Yeah, no, I think that's another really hard question to think about. I definitely think there's a lot of functional redundancy in microbial community. I mean, these are incredibly diverse systems, and I think they compete with one another quite a bit, but I definitely think there's some functional redundancy there. In terms of like their sort of positive impacts on plants, I typically think about, yeah, plants are important, but I'm thinking about the other organisms that feed on these microbes. And so I definitely think, yeah, I would say a good portion of them would be very beneficial. And just thinking about in general, maybe it's the ones that are most abundant, you know, I'm thinking about the ones that are most abundant would probably be the ones that have a bigger impact, but that's like always context dependent, right? And measuring abundance is somewhere really tricky, especially when you're doing sequencing stuff as well. So it seems very easy to say, but it's also hard to kind of to know exactly which one is we're really driving that in this case here. I'm always a big proponent of thinking that the rare species are rare and they probably don't have really huge impacts, but you can always point to like exceptions where that's not the case. So, but that's the best answer I have. Right? Yeah, I think it's a difficult question.
I know, though you should be, you're not all soil ecologists, so I will try to change directions a little bit, yeah.
I'll ask you a question. So, this is relating back to like a recent study that you had. I think you talked about it near the end of the talk yesterday about invasive root traits and how they affect microbial like homogeny in the soil.
Right.
And so, what I was just curious is that for that study, it was a very broad study because you were just looking at like invasive plants in general, which varies greatly, like just within that group. So I was just curious as to what you think, I guess, the impacts of that study are in terms of like how true they could be across, like, I don't know how true could it be for like a cheatgrass compared to like garlic mustard, buckthorn, stuff like that.
For sure.
Yeah.
No, I appreciate that question. And I think that you're absolutely right. I think there's huge variations across invasive plants, and think about the category of invasives from like woody to versus thinking about the different systems that they invade from wetlands to forests and so forth. And actually for that particular paper, I think that's what makes that the strength in some ways is that even when you take into account the ecosystem, the type of plants and all that, you see this homogenization effect. And so it kind of points to that a lot of these, even though they may be different functional categories, they are really impacting microbial communities in similar ways in that case there. One of the things I would say that just naturally that, you know, there's a lot of work that's shown that invasive plants usually have what we call like cheap roots. So they're really fast, they grow, they break down very easily, they acquire nutrients and can monopolize the nutrients. And so they often correlate then with a shift from like from a fungi-based soil to a more bacteria-based soil in that case there. And that study kind of really shows that as well, is that you do get more cheap roots with invasive plants more broadly. In terms of like the individual comparisons, yeah, it's really hard to think about that. But you know, I will say just in general, when you kind of lump invasive plants as this broad category, you do see that they change. They look very different when you compare them to native plants in the case there. But that being said, as another side, I'm hoping actually to kind of resolve that a little bit more. And I absolutely agree that all of these invasive plants are different, but I bet you can find some similarities in like the category of traits. So allelopathy as a trait, nitrogen fixation, plants that have like, invasive plants with like really cheap, fast growing roots probably have a different impact. And you can think about those as being context dependent as well, where nitrogen fixation probably doesn't really benefit you when you're in a very resource rich environment. But if you're in like a tundra where resources are very limited, that nitrogen fixation trait as an invader probably can do some wonders in increasing invasion success. And so I would like to kind of build out, a framework of how just these unique traits and try to find commonalities about how they can impact invasion success, but then think about their sort of impacts on the soil ecosystem.
Yeah, yeah, for sure. And then just a follow-up question with that too. Because another thought I had is that like all these plants that are invasive here, they're native somewhere else.
Yeah.
So I don't know if you've looked into, if you've seen any studies. If you know, do these invasive species and how they like alter the soil communities here, do they have those same effects in your native range, or I don't know, is there some difference there?
Yeah, no, it's cool because like there are some studies, some really clever studies done by like Reagan Callaway at like Montana, University of Montana, I think it's Montana. Actually working with garlic mustard in this case, and it's actually kind of a very cool idea. It's looking like garlic mustard, where basically you had garlic mustard plants from like Europe and compared them to like garlic mustard plants that are now actually invaded in North America, and had sort of translocation of soils and seeing what the impacts are. But it's something about the invaded sort of genotype that really it has really pronounced impacts on the sort of novel ecosystem, whereas in its own native range, it doesn't have the same influence. And I would envision that a lot of the invasive plants probably show similar patterns where in its native range, for whatever reason, they're not really problematic, probably from like natural enemies or pathogens in the soil or something like that. Whereas when they introduce into this new system, that all those things are no longer a problem, they just have a competitive advantage over native plants.
Just to follow up on that specific to garlic mustard. So does it mean that when people are trying to restore an area that had garlic mustard in it, the only way to be successful is to reintroduce the microbes in the fungal community back in, which you just said maybe doesn't actually work in reality? Or how does, just since I know it's a species, a lot of research has been done on.
Yeah, I don't know the most recent approaches for trying to restore areas that have been invaded by garlic mustard, but I do know, if I can recall correctly, when you remove garlic mustard, especially in heavily infested patches, it takes about like 7-8 years to get that patch to look something similar to something that was pre-invasion. And so it's the fungal community takes a long time to restore. And so you can envision that if you have this eroded fungal community, like even after you remove garlic mustard, it probably would take a long time before you kind of get it to a point where native plants can actually emerge and do well in those soils. But my understanding though is that, at least back in Chicago, when you had issues with garlic mustard invasion, you certainly try to get it in the small rosettes, but certainly before the set seed. And yeah, and in terms of like the impacts of the soil, yeah, we really haven't figured out a way to kind of restore it outside of just like it just takes time to get it back to where you. Yeah, well, I guess if we can inoculate soils, maybe we can accelerate that process. I'm thinking that.
There have been a number of studies in the Midwest. I don't know if it's been with garlic mustard, but a number of soil translocation studies in the Midwest where just add a little bit of topsoil just as a native inoculum. So some in Europe too, to early, late successional sort of soil translocations, a little bit of inoculation. And there are examples where it has worked. Yeah, it's really helped things reestablish, but I think there's a lot of unpublished studies where it didn't work too. I'm talking to people.
So I have a question about just like your bacteria and fungus in general. I'm not a soil scientist, so maybe this is obvious question or kind of that, whatever. But, so it's clear that you spent a lot of work looking at like these free living bacteria and fungus that are in the soil. I'm curious about like the endosymbionts, maybe within those higher trophic levels, the predators and these other various invertebrates, and if you think that plays any, like role in the community, if like, like I said, I don't know much about soil, but I can imagine that some soils are more nutrient poor and some are have different nutrients that maybe like these invertebrates aren't getting their like full amino acid compositions. Maybe they have endosymbionts that kind of fulfill those like lacks. I was wondering if you know anything about that or can speak generally. Endosymbionts and how they might relate to your free living bacteria or.
Yeah, no, that is, trying to think of, I know, any obvious ones in the soil. It's most likely gonna be some arthropods and invertebrate species in general that may have endosymbionts. And they, they probably are protected against predation or, think about drought, soil conditions and things of that nature. But I can't think of any, off the top of my head, any obvious examples of that, at least regarding things that live in the soil. I mean, obviously the aphids and other arthropods above ground certainly have benefited from that, but I can't really think of anything below ground. So it'd be fun to look in the literature to see if that is a thing that arthropods have.
Yeah, I have no idea. That's why I was wondering if you knew, or maybe it's undiscovered or just not looked at, or maybe it just doesn't exist.
I would put my money on it. There's certainly some cases in the soil where you have endosymbionts of like arthropods and vertebrates more broadly, but I don't actually know any that I've worked with that would have such a thing that that's pretty nice, but I don't know. But I will look it up now. Now you got me curious to see if there is something that I'm missing on that. I don't know. Yeah, there is.
And not insects, but I know fescue has an endosymbiont fungus that provides herbivore defense, and that one's been pretty widely studied. And then I do know also there's, I think it's an orchard. There's an orchard insect that orchardists will introduce a entomopathogenic nematode that attacks that is a biocontrol for it's a citrus bug. But as far as symbionts, yeah, I'm not.
Thanks. Sorry, I know that's kind of random, but.
No, it wasn't. I appreciate that.
All right, we can switch gears here a little bit. You guys, people have sent in some questions, a little more broader. How about, how'd you get into ecology?
Yeah, I definitely fell into it by accident, I think, I grew up more in the urban environment and didn't even know like ecology was a real career until my freshman year in that's in undergrad, and I actually had like a professor. I took one of his intro level biology courses, and he was looking for some undergraduate research assistants to work with him for the summer, and he kind of recruited me to work with tiger salamanders. So I stayed around for the summer. We got a chance to go out to like the local pond, and it was like the tiger salamander that had tadpoles, and you going out there and had the seine and getting muddy and grabbing all of these the juvenile tiger salamanders, and to me it was a lot of fun, to be honest. I know some people like, you got all muddy, and I'm like mud all the way up to my shoulders more or less. And we were able to take those back and run some experiments to understand how crowded conditions can change how fast or slow they metamorphose. And so that was kind of the first really ecological sort of like experience. So I was like, wow, this is really fun. And that kind of really shifted my gears towards doing ecology. When I was going into undergrad, I actually thought I was going to be a veterinarian. I always liked animals and figured that, oh, that would be kind of cool. Like I can like save animals for a living. But my sophomore year, I had this animal physiology course, which I thought was going to be really fun. And it turned out to be traumatizing in some ways. I went to a small liberal arts. I went to a small liberal arts college, and so what's kind of cool about this class, at least initially I thought it was cool, is that we had each of the sort of members, participants in that class, we got a female rat that we had like, to me, I thought it was a pet, but basically it was a lab rat more or less. And so, but what we had to do was like take out the ovaries of the female rat and then inject the rat with unknown hormones to try to track it through its like menstrual cycle more or less. And so you can kind of, the idea was that once you remove the ovaries, you can kind of inject it with this hormone and you can kind of see what hormone you injected it based on like where they are in the menstrual cycle. You can look at the cells of the vaginal cavity, which, you know, it sounded like it was a lot of fun, but in order to actually take out the ovaries of the female, we actually have to perform surgery. So we have to anesthetize the rat, put it to sleep, we have to cut it open, remove the ovaries, and then stitch it back up. I don't have a stomach for that. I almost passed out several times. I was the last one in class. It was quite traumatic, and that was the thing. I was like, yeah, maybe I won't be. I was like, it's okay. I still like animals. I'm not doing any surgery. I can still do that. I don't have to do any surgery. Fast forward, so I'm still, you know, my rat at that point didn't trust me, but I had to do it. So I built the trust, and then after a while, it trusted me again. It got very excited seeing me, and it's like my friend after a while. And then at the end of the class, I had to put my rat down. And then I was like, that was like very hard for me actually. And I was like, you know, maybe I'm not built to do that. I know those are things you have to do. So that combined with like, you know, working with salamanders and then going into my senior year, my junior going into my senior year, I actually had the opportunity to do an internship for the Environmental Protection Agency in Oregon, Newport, Oregon. And that's what really solidified my interest in doing a PhD in ecology, because I got a chance to shadow EPA scientists for a summer and spent a lot of time outdoors looking at how sort of, I guess, urban impacts on salt marshes in that area. So we ran some experiments. I couldn't believe that he spent this entire summer pretty much outdoors, checking on experiments and taking samples. And I was like, that's what I want to do basically. And so that was kind of the solidifying sort of experience as like, yeah, ecology. That was a hand, maybe?
Yeah.
Yeah.
This is kind of like switching gears a little bit, but I guess, any advice would be great, but I always found that like for me, my greatest struggle in science is like the writing. And I think that's like so much of it too.
Yeah.
And obviously you have like written papers in some pretty great journals. And I was just wondering like what your approach to science writing is and like what advice you would give to grad students.
Definitely, yeah, and I really appreciate that question. And you're absolutely right. I think to be a successful scientist, you would need to enjoy writing and kind of see it as a way to communicate all the cool stuff you get a chance to do in that case, you know. But honestly, my approach to writing is, you know, different stages. I think that, you know, put as many words on paper as you can. So I feel like, you know, there's a tendency, especially thinking about my own grad students, they want to have these perfect first drafts. And what happens is that it really inhibits their ability to actually just put their ideas out there. And so it actually, I think, it takes a lot more time to do that. So just kind of word vomit, you know, as much as you can. Don't worry about how it sounds or how it, you know, how it may look, but to just try to get all those ideas back on paper. And then you can always go back and refine and be okay knowing there's going to be several drafts before it gets to a point of quality in that case. Yeah. But honestly, just about everything that you do from like lab work, field work, or exercise or skills that you're trying to build, in order to actually be really good at it, you just have to continue to work at it. And I think that, so being intentional about, you know, reading books on how to write scientifically, think about sentence structure, think about paragraph structure, think about the narrative that you're trying to build. And really take it as a point that like the more you do it, the better, and certainly get feedback from the broader community. So I feel like I'm always thinking of like my writing as a product that I'm constantly working on. I never feel like I get to a point where my writing is where it needs to be, but I'm always looking for new ways to develop my skill as writing. And so that would be the last thing I would say is that just understand it's always like a work in progress. And so don't be too hard on you if you feel like you're not where you want to be, but just know that, you know, at the end of the day, it's fun to be able to put your ideas, especially if you got, you know, some cool experiments. It's good to kind of get that written up and for people to actually know what you did and that, you know, it's just a practice that you just want to try to, I guess, perfect. Peter, Alan, do you have some thoughts on that?
I look at the data and then I come up with my story and I write the abstract. And I'll write the abstract 100 times until it's a smooth, concise story. And you have the whole story arc in your abstract. And that's before then I'll write the discussion and then I'll write the intro. And then you don't talk about anything in the paper that's not in that abstract. The abstract tells your whole story, and don't throw in some. I love appendices because, you know, grad students always have 50 figures that they want to show, but you should only show like 2 or 3 figures. Go look at a bunch of papers, read 100, look at 100 papers and just do a paragraph counts. There's 4 or 5 paragraphs in the intro. There's 6 to 10 in the discussion. Like, it's pretty rigid, like, and you don't have much time to say anything. So get your story tight. There's lots of stuff you're going to cut that you want to tell that's not going to be included. But work on that tight story that's easy to tell, and then only include information that tells that story. Peter? Everybody has a different approach though, so. Talk to lots of people, different approaches work for different people. But writing, rewriting, practicing.
Yeah, I guess I'm thinking about the part of your question that you said you don't enjoy it.
Oh, no, I was just saying it's like hard. Like,
Is it, but yeah, I mean, it's good, it's always hard.
yeah.
Even when you're good at it, it's still hard.
It doesn't just happen.
And, but I still enjoy it, even though it's hard. And maybe it's the process of getting it from the word vomit stage to that polished stage that I enjoy. Sometimes I think of it as like building a brick wall. You know, each sentence is a brick, getting them in the right order. Seeing it sort of take shape. I don't know, but I think if you want to be a researcher, you have to learn to enjoy that process, otherwise your job would suck. And maybe, you know, there's probably some people that it's just that writing is going to be the reason they don't end up in a research career. So, you know, if that's where you want to go, I think figuring out everybody's got advice, everybody's got a different process. I was curious which books you've liked, but try different things and try and figure out how you can learn to enjoy that struggle. That's sweet.
It's, I can write a draft of a paper in a day, right? And I will spend 2 or 3 months rewriting it. I will rewrite every sentence literally 100 times before the paper is ready to go. It's just lots of revision to make all the pieces fit.
Yeah, and I think for me, I've always felt like writing was more of a natural way to communicate. Like I know humans, we have to speak and all those things. And I feel like when you actually write, you can really sit down and really think about like crafting the sentence and thinking about the story arc and really craft a story. I think to me, that is very satisfying. I do have a lot of fun with that. But it wasn't like that in the beginning. You know, I definitely was like, man, this is like taking forever. I've been looking at this page for 4 hours and only got like 3 sentences, you know. And so, but it is something that I think that, you know, to Peter's point, is like you have to learn to enjoy it. But it is like extremely gratifying when you're at that point where you learn to know that it is hard, but you can, you know, really get some good communication out there about a certain topic that could be, you know, really important, at least important to you. And I think it's really satisfying to get to that.
And maybe one way to enjoy it is not to panic. at how hard it is. And I mean, and I think the more you do it, the more faith you develop that, oh yeah, it sucks right now, but it's gonna get better. I'm gonna get it to where I want it to go. And everybody will arrive at a different process for doing that. I'm listening to a book right now about productivity in general, how to get quality work done in this world full of distractions. And he opens the book with an anecdote about, so John McCary is a famous nonfiction writer from, because he's old now, but it was a New Yorker writer for 50 years, published tons of books. And apparently one of his first books, he spent like a year doing all this research and all this material. And then when the time came to start writing it up, he spent 2 weeks lying on a picnic table in his backyard, looking up at a tree, just thinking about how to organize it. And eventually after 2 weeks, he figured out, oh, there's this one character who is like connected to all these other parts of this topic. And he could tell the story through that one character. So then it still took him another year to write the book, but he took that he was okay with basically doing what looked like nothing for a long time. Until he was ready to write. And I find, so I do a lot of outlining, and I often don't actually try and write in complete sentences until my outline has stopped changing, until I know what I want to say. It's really hard, I find it. Some people use like writing as a way to figure out what they want to say. And I tend to work on it more when I'm going for a run or something, and I don't actually sit down to type out the full sentences until I'm pretty sure what I want to say. So outlining is one way to do that. Like when my outline stops changing every day. Like, okay, I think I can write the introduction.
Another little trick I use sometimes is, I think people, especially grad students, have a sense from reading papers that they find difficult to read, and they think scientific writing is complicated and hard to read. It's not what you want it to be. You want it to be easy to read and easy to follow and have a simple story that you're talking. And I will often, actually, I'll be on a bike ride and I'll pull out my phone and I'll just do a voice recording or something, and I'll just say it out loud, and then I'll transcribe what I said, because you speak much more casually than you write. It's a good little trick. But okay, so on this topic though, we've got a couple questions here. Then, how about do you have a favorite? These are kind of related. A favorite paper, and what do you find makes a good paper?
Ooh, great question.
I mean, other than mine.
Yeah, so actually, great question. So, I mean, this is some ways, I don't know if this is cheating or not, but actually like Adam Smith has this like classic study that shows like non-consumptive effects of spiders, I think on grasshoppers in this case. One of the things he did in this experiment that was very clever, I don't even think it was, the paper, I wouldn't say the paper was like great in terms of how it was communicated, but it just was kind of a very clever experiment to actually look at the non-consumptive effects of spiders when they glued the mouth parts of the spiders and compared it to like actual spiders that can actually predate upon these grasshoppers in this case here. And what he was able to show is that just by the presence of the spiders actually suppressed the activity of the grasshoppers and actually had this sort of cascading effect on the vegetation in that case there. But to me, it was like such a clever experiment where you can, envision that, questionable on the sort of like ethics of it, but.
I was there when he was doing that experiment.
Oh, he was there? That's,
In Wales, yeah.
yeah. But it was like such a cool experiment. It's something that I often like when I'm, preaching to the grassroots, I'm like, these are sort of the really elegant experiments that may seem very simple, but they can actually really be very important for like pushing the science forward. And so that I think that that's like one of the studies that kind of like stick out in my mind. It's like, like what's really clever in terms of like pushing the field. What makes a good paper? I definitely think I've been really thinking about like, and I think that there's this tendency, as you mentioned, like grad students feel like the harder the paper to read, and the more scientific it becomes. I'm like, no, that's not how it should be. There should be a paper that is like very well written, easy to understand, and has all the components that, you know, you would expect to have. And so thinking about like a show or a movie where, you know, the intro is setting the stage for all the characters that will be important, and having sort of a set of very clear questions that they are trying to investigate. All the figures in the tables correspond to addressing those questions. And then they have a discussion that really describes what that means in the sort of broader context, and having very little, you know, very few sort of distractions along the way, and really having a tight story in that case there. And I actually do think that like paper should be fun to read. I know we kind of get in the mode of thinking that scientific paper should be bland, but I think that like, you know, thinking about word usage and how you sort of craft the argument and making sure there's a logical flow, like those things are what I really enjoy reading in papers.
And just a question that's, I guess, somewhat related to just like scientific literature, but more so on like the project design part of it. Like especially now, I mean, like every single day, more studies come out and there's just a plethora of knowledge out there.
Yeah.
And like when you're designing your experiment, you often want to try and find like these gaps in the research that you could hone in on and like research. So I guess, do you have any advice for finding those gaps in the research?
Yeah, I definitely have some thoughts around that. So I think there's like two parts to it in my opinion. So one is like, what is like your natural interest, you know, and thinking about like what really gets you excited about the type of science that you do, and really having that as like your guidepost for the type of research that you're going to get into. And then from there, I think that once you kind of have an idea of like what really gets you excited, start reading literature around that particular subject matter, certainly reading like more recent literature. If there's like a recent review paper, they usually kind of highlight some of the key gaps in that way. Or if it's like some outstanding question no one has really addressed yet, and thinking about approaches that maybe you can implement to try to sort of fill those knowledge gaps. Those are kind of the easiest ways I kind of think about, you know, how to actually sort of like go about trying to address some knowledge gaps. Thinking about well, the ones that are pretty obvious and no one has really addressed are the ones that, like, you know, as people are publishing and coming up with topics or studies that are pushing the field forward and trying to see if you can kind of get ahead of that. But ultimately, at the end of the day, it should be what your true interest is, and really using that as your guidepost, I think. And then you can always find a map that's got them. Do you have similar approaches?
For designing experiments or for finding the topics?
More so just like finding topics, I would say.
I find when you're just doing literature, like just reading a bunch of papers, it's just somebody spent a lot of time writing a paper and like making it engaging and interesting, and it just always like I'll just every morning usually when I wake up I'll tend to read 100 titles, 10 abstracts in one paper, right? So every morning I'll wake up and I'll read 10 or 20 or 30 titles and 1 or 2 abstracts, and then every couple of days I'll read a full paper, and like, but whenever I read those, I get a flood of new ideas, and often another way that ideas come up, and somebody had a question about how you learn modeling and build models, but if you try to when you do try to plug parameters into a model, you know, and you try to look for the data you need for that, you often realize, oh, nobody actually measures this. Nobody actually measures this thing that we really need. And you're like, well, that's an obvious thing we need to do. Like, I do a lot of root water uptake. It's like how the plants get their water. Like, it actually nobody knows. It's really hard to figure that out. And so it's like I've been doing that for 15 years. So. Yeah, so I do just a lot of reading, just read. I think you'll get lots of ideas. But I think we've just a couple minutes here. Anybody have any final questions?
I want to ask a question about So there's not many black colleges. And I'm guessing that you get lots of pressure or opportunities
Mhm.
to work on diversity committees. Yeah, is this a burden? Is it not? Someone like me isn't gonna get as much pressure to represent. So how do you navigate that?
Yeah, that's a really, really good question. And honestly, I definitely think there are certainly a lot of opportunities in some ways, like overwhelmingly a lot of opportunities. And I think certainly what happened in 2020, there's like at least there was some people that realized that there are some disparities between Black communities more broadly, and certainly was similar to that, like ESA, put a task force together and having as many of their certain Black ecologists as they could with opportunities, which I think is they're good opportunities, but I think that for the individual, it's really about understanding that, time is finite and you have to protect the things that you really care about, and especially as like, junior faculty, thinking about the things that really matter towards like promotion and tenure. So protecting that time, making sure that I'm doing all the things that I need to do for my publishing, submitting grants and all those things. But then also understanding that I feel like I do have a duty, as one of very few Black ecologists to try to make the field more diverse and really create space for individuals with similar backgrounds, similar sort of, dimensions of diversity, outside of being Black, but also, people from low-income neighborhoods or all those different factors, and thinking about, am I creating space for those individuals as well? And so the way I kind of approach that is honestly, if I feel like I can, for one, if I have the time for it, if I have the time for it, is it something I'm passionate about? Is it something I actually do, that I do think will make a meaningful impact? And then is this the sort of brand that I want to build for myself, And so that's kind of the sort of lens that I use to kind of decide when someone approaches me about, you should be a part of this committee, or you should be engaged in these activities, we want your thoughts. And so I'm like, do I have the time? Is it something that I care about? And is it supporting the brand that I think I want to build? And then usually that helps me sort of make a decision on if it's something that I can agree to, or something that I just feel like it's just not a great time for that. I would say my approach. There's certainly other colleagues that have very different approaches. Some who feel like, and I totally understand it, they're like, well, I get that I represent this sort of dimension of diversity, but it's not their duty to do any of those things. And so some people like just, they don't have to care for that. And then I have others who like, that's all they do. They wrap their research program to be able to actually pursue those pursuits, and so they like fall in, So I feel like I'm somewhere in the middle of things there. But I do think it's a core part of my program. I'm always thinking about ways in which I can make more space for people with diverse backgrounds to feel like ecology is a place for them. So I appreciate that question.
A quick follow-up, I can't get a minute.
1 minute.
So can you give us one example of something you're doing in that area that you feel really good about? There's an obvious yes,
Yeah.
and then a request or invitation or something that you turned down, didn't feel like it was a good one.
Yeah. One that was an obvious yes. So I'm technically a part of this, they called the EED language project. And so I had some colleagues reached out around this idea that, the language we use in ecology, a lot of, the founding fathers, came up with some really racist, sexist terminology, some of which we still use today, thinking about species names, for instance, a lot of issues around that. And someone reached out as part of the group that they're trying to develop. And the idea is to try to get a group of scholars in EED and really start to think about how can we make the field more welcome and inclusive by really interrogating the language that we use in the field of EV more broadly. And to me, that was like a pretty obvious yes. I'm like, you know, we published papers, we got, you know, submitted grants around this, and it seems to be, it was like the perfect sort of opportunity to still do scholarly work, but also actually make a more positive impact in that area. An obvious no. I say no a lot, so I'm trying to think about when was the last time I had to say no. Well, one of the things I do find quite annoying, honestly, and I get where this is coming from, but there is like opportunities where you know, people are trying to diversify like the editorial board, or thinking about diversifying elements of, you know, like a committee or something like that. And I can, you know, and actually there was like actually the it was some society, they wanted me to join the editorial board. I think it was society. Yeah, I'm like, I don't even identify with your society. I'm like, why are you reaching out for me to join your board member or whatever? I'm like, no, like I don't even it. And I knew exactly where they were coming from. Like, well, they need to diversify folks who are making decisions for that society. And you know, my name came up, and I'm like, I know nothing about you, so that was like obvious. Yeah. So, but yeah, but those are like some of the. Yeah, I get some invites all over, like, you know, seminar visits, you know. I'm often, if it's a place that I'm really interested in visiting and it's cool people, I'm most often going to say yes to that, but certainly turn those down as well. And the ones that, yeah, I don't know, there's some really weird ones, like, you know, that it turns out to be like a semi-interview, and I didn't know I'm interviewing, and all of a sudden it's like, hey, what do you think about institution? I'm like, this is just a research seminar. What do you mean? So I've gotten that as well. So it's
All right, let's thank Dr. McCary.